Greetings, one and all. And in Darug, the Sydney language, Warami, which means good to see you. I begin by acknowledging the First Nations around the world, wise custodians of the earth over tens of thousands of years. May we listen to God's guidance, which they have been cherishing through the eons. I am Doro Kostake, an associate professor of theology at the Sydney College of Divinity, in which capacity I chair the Theology Research Network, TRN, as well as director. It is my pleasure to host the last TRN ISCA seminar this year on the topic Religiously Human in a Techno-Scientific World, and to introduce today's speaker, Dr. Hermina Nedălescu. Hermina is a Romanian-born Orthodox Christian and a neuroscientist at Scripps Research in San Diego, California. She holds a PhD in biomedical science from the University of Antwerp and is a theologian in training in the Center for Theology and Natural Sciences at the Graduate Theological Union, Berkeley. Her scientific work is concerned with the neurobiological control of abnormal behavior and brain functions relevant to human psychopathology, focusing on substance use disorders and other addictions. Currently, she develops a theological anthropology at the nexus of the Orthodox Christian tradition and addiction research. She is a member of various scientific and theological associations, such as the International Orthodox Theological Association and the Society for Neuroscience. She is also an instructor for Step in High Incorporated, the high-based organization funded by the County of San Diego Behavior Health Services Department for the training and support of clergy, pastors, and health providers who minister to people suffering from SUDS, other psychological addictions, and mental illnesses. Hermina integrates her scientific expertise with artistic expression to explore and communicate concepts related to structure and function. Overall, her wish to bridge the gap in science, theology, and the broader public through art and dialogue, making complex scientific ideas more accessible and engaging. And I will have the honor of responding to her paper. Now, let's turn to Hermina's presentation, Uncovering the Invisible Universe of the Brain at the Crossing of Neuroscience, Art, and Theology. The paper will run for about 50 minutes, after which I'll be giving my response for about 10 minutes. Before we do so, however, please make sure you have your microphone switched off for the duration of the seminar. Equally important, if some of the attendees do not wish to have their faces appearing in the video recording and the print screen photos, please keep your cameras off. Hermina, the floor is yours.
Okay. Hello, everyone. Can you hear me okay? Perfect. So it was breaking out a little bit. Just let me know if it is breaking out again and I can speak slowly. And Doru, thank you for that introduction. Are my slides okay? You don't see the notes on your side? Okay, perfect. Well, thank you for inviting me to give this talk. I'm delighted to be here with you. It's going to be about the brain, and then I will put my theologian hat on and try to make some connections from my scientific observations. So let's, one moment. Working with two screens, I'm sorry. I'm trying to minimize, okay, my face. All right, here we go. All right, so the title of my talk is Uncovering the Invisible Universe of the Brain at the Crossing of Neuroscience, Art, and Theology. There is much we can gain about who we are if we work at the intersection of these three disciplines, because neuroscience allows us to see inside the brain, and art actually provides us with a palatable way to describe what we see under the microscope. And then theology enables us to be able to go beyond the limits of the science. So the outline of the flow of my talk is as follows. I will be talking about showing the complexity of the brain by looking at the structure and function relationship. And this will be mainly from microscopy images. One moment, I have something not, okay, I need to move something on my screen so I can see. All right, then I will, really throughout the slides, I will be presenting my central question and position, which is interaction gives rise to existence and shapes our reality. And then I will shift into speaking about the model of the Trinity and how the relational interaction between God, Christ, and the Holy Spirit is also related to us as human beings. So, all right. So the brain is, without a doubt, one of the most complex structures that exist in the universe. It allows us to think, to imagine, to reason, to feel various sensations, and to act. What neuroscientists are trying to understand is how the brain allows for all of these operations to take place. The complexity of the brain doesn't just depend on its structural architecture and its various elements, but rather on the immense quantity of interactions between these elements that constitute the brain. To give you an idea of this complexity, the brain contains about 100 billion neurons and trillions of synaptic connections, which are the point of contact between neurons. In addition, there are about 40 to 130 billion glia cells, like the glia cells you are seeing here. These are 2 glia cells, which provide support for neurons. So ultimately, the brain, it's important to know that it's ultimately a calculating system. It is architecturally constructed of neurons that enable us to calculate, compare, and make decisions based on these calculations. For example, if we plan to jaywalk across the street, we need to be able to calculate the distance between the car and our body to make sure we can cross safely. However, sometimes the brain miscalculates, and this happens a lot more than we would like to admit. So how does the brain and the brain circuits provide these calculations and comparisons to support our actions? One of the key components to understanding the brain is to study its architecture, because by learning the architecture, it gives us some insight into the function. So much like the Trinitarian doctrine was fervently discussed about 1,600 years ago, a lively debate about 125 years ago led to something called the neuronal doctrine. So we have some doctrines as well in the sciences. And here's what this neuronal doctrine said. So there was a debate between these two individuals. One was Golgi, Camillo Golgi, an Italian, and Ramon Santiago y Cajal. Golgi had developed a clever silver chromate staining that enabled him to be able to see the different neurons. And he came from his observation, he thought that the neurons were connected with each other in a type of continuous reticulum, much like the blood vessels are. Then Ramon Santiago y Cajal came along, and he was actually an artist. So he interpreted the staining to mean that the neurons are actually individual units within the brain, so that they're not continuous with each other. So here's what the two saw. Golgi saw this continuous reticulum, like blood vessels in the body, and he said that there is no space between brain cells, and that fluid can flow uninterrupted from one neuron to the other. Meanwhile, Cajal said that actually there is space, and this is how he represented it here at the synapse, and that neurons are actually individual units, and they are contiguous with each other, but not continuous. We now know that actually they were both right, because there are something called neuronal nets. But at that time, and for the past 100 years, it's Cajal basically won. So we are driven by Cajal's idea that the neuron is an individual unit. And I want to share this quote with you just to give you an example of how beautiful the brain is and how we can learn so much from it. So he writes in his memoirs, "To know the brain, we said to ourselves in our idealistic enthusiasm, is equivalent to discover the material of course of thought and will. Like the entomologist hunting for brightly colored butterflies, my attention was drawn to the flower garden of gray matter. Gray matter is the neurons in the brain, which contains cells with delicate, elegant forms. The mysterious butterflies of the soul is what he referred to as the neurons, with beating of whose wings may someday, who knows, clarify the secret of mental life. Even from the aesthetic point of view, the nervous system contains the most charming attractions in our attractions." Then he adds, "In our parks, are there any trees more elegant and luxurious than the Purkinje cell from the cerebellum or the psychic cell that is famous, the famous cerebral pyramid?" So a lot of the brain neurons look like trees. And I will show you what he saw. So this is what he's talking about, the garden. And the famous Purkinje cells are here in the top left. These are stellate cells. But I want to show you how important the artistic interpretation is. He was an artist in addition to neuroscientist. So this is the actual tissue that he saw. This is the Golgi stain. And you can see how dark it is. But he managed to extract from this. So he was able to, with art, we can see these nuanced features, these, beautiful arborization of the Purkinje cell. The stellate cells are here. The basket cells are here because they form like a basket. Then the little ones here are granule cells, which are the most numerous cells in the brain. And then we have the Golgi cells. So this is what can be done. Now, the brain is hard to study. It's a challenge because we have to be able to have ways to study the synapses from the nanometer level all the way to the long axons, which are in the meter scale. So it just spans such a huge this whole metric system here. And it's hard. We need different tools to see the synapses. We need an electron microscope to see these long axons that project from one side of the brain to the other. And especially, they're long. We need different method to be able to see them. So that makes it challenging. But the methodology, the technology has gotten better. We have the equipment, the microscopes to see, and then we also combine with genetic methods now. We no longer use the silver chromate that gives us that dark stain, but actually someone was able to extract the fluorescence, the genes that encode fluorescent proteins in jellyfish, and they were able to put that into an animal and create these transgenic animals. So here you're seeing all the Purkinje cells lit up. It's a very zoomed out region. I'll zoom in a minute. But we are able to express fluorescent protein from the jellyfish now under the control of certain cell types. They each have a special promoter, and now we can light up these cells. Another method that I use is I inject viruses which can be used as vehicles to inject these fluorescent proteins so that I can light up my cells of interest. So here's a cross section of the Purkinje cells. They're actually green under the microscope, right? So then we shine a laser on the tissue, and there's a CCD camera that captures all the excited photons, and then it creates this image. And you can see the arborizations here. Now, it's still hard to make sense of it, but if we go in with digital methods, we can actually draw each one of them. And now we have access in 3D space to all of these neurons, and we can do statistical analysis and really study the architecture so that we can understand the function a little bit better. So here's how it works. Here, this is a volume, in the brain of a rodent. Purkinje cells are lit up with a fluorescent protein. This is captured with a 2-photon microscope. So you get a nice chunk, so you can begin to see what this population looks like. But if you really want to study it, you have to be able to draw them. And this is what I do. Once you begin to extract the data, like in this kind of art form, you can appreciate that they're planar. They're kind of like coral. And there's a reason for this. The reason being is that they spread out their arbors in a planar form shape because they actually receive about 200,000 inputs from this other cell type that is sitting underneath and sending its fibers up. So in order to capture as many parallel fibers as possible, they are structured in this fan-like way. And then we can start asking another question, and that is, what is the relationship with the neighbors? What is the position that each of these trees are in relation to each other? And that was a lot of my work that I spent on doing my doctoral studies. So we can, again, like I said, if we draw them, we extract this data from the brain, we can not only show the public and talk about it, but we can also appreciate the structure. There's a organization. It's not random at all, the placement of these cells. So I'm going to go on to talk a little bit more to give you the bigger picture of where these cells are coming from. So if you imagine this box to be the cerebellum, I'm speaking about the cerebellum right now, which is a brain region that's very important for motor movement, for walking, for playing piano, for grabbing a cup of coffee, even your eye movements. It's important for all the movement. And so that Purkinje cell that Cathal spoke about, that beautiful cell in the garden, is here. And these are the representations of those parallel fibers. So 200,000 of them usually contact one of these arbors. And that's why they are flat, so that they can capture as many as possible. Then, of course, there are other inputs coming in and other outputs. But I want to focus on this particular one from the locus ceruleus just to show you. This one provides, it's a system that enables us to be alert so that we don't fall asleep. It's called the noradrenergic system. It's important. It's basically the arousal system. It helps us escape from a rattlesnake or stay awake during a boring talk. And here's what it looks like in there. So these are the cell bodies of those Purkinje cells. And now we see this net-like structure. And the dots are actually where the neurotransmitters are. So again, I drew these digitally so I can extract them and study them more carefully. In fact, I was looking at what the distance was between these dark black sacs that contain neurotransmitter in across different conditions. I won't go into the conditions, but it gives me access to study the structure a little bit better and connect better with the behavior of the animal. This is called a global system, the noradrenergic system. It's not a point-to-point system. It's not one neuron connecting another neuron. It's a global system in the sense of it casts a net over all the neurons that are in this area, so that this neurotransmitter can be released and available to all the neurons at the same time. Okay. Oh, all right. So that's the noradrenergic system. Now I'd like to show you a little bit what it looks like if we were to follow. So a neuron has a cell body, a dendritic arbor where the information is received, and then it has an axon, which is the part of the neuron that sends information out. So let's look at the output of these Purkinje cells. What does it look like? So here we have, if I can draw your attention to the bottom right, we have these Purkinje cells that are sending these axons, and you can see them that they form these bundles. There's so many of them. In the rodent, there are 200,000 Purkinje cells, and they send these axons, and then they they branch out like spaghetti over here onto another set of neurons called the deep cerebellar nuclei. And here's what those look like. Red is the Purkinje cell axons, and they are converging onto these green cells. So there are, in the rodent, there are about 200,000 Purkinje cell axons, which then branch and converge onto 18,000 cerebellar nuclei. You can appreciate it a little bit. There's really massive connectivity here. So again, if we follow one of these Purkinje cell axons, we can begin to understand where the connectivity, how the connectivity patterns happen. Does it happen on the cell body, which would be a much more potent inhibition, because the Purkinje cell provides an inhibitory, it's inhibitory information in the sense of it shuts down the neuron that it's talking to. And then here you have on the dendrite, another connection there. This is just one salient feature. There are a lot more connections, actually. Let me show you in the next slide. So these are 3 examples of the deep cerebellar nuclei, and you can see how the connections wrap around the cell bodies or also the dendrites. So these neurons aren't as beautiful as the Purkinje cell. They're not planar, they are actually very spherical. And not all the neurons are beautiful. But here, sometimes I use these images from the microscope to show people a window into the brain. So here you see a cross-section of the cerebellum with those Purkinje cells, and I color code them to make it more artistic, but it gives me an opportunity to describe the structure of the cerebellum in art form to people. So I do exhibits with these images. So here you can see the cells, they actually are aligned in a monolayer. You can appreciate the structure. It's not random at all. And you can see also, if you look really close, you begin to see the spines. And the spines, spiny protrusions, are the sites of synaptic connections. And then you look at the axons, which the information is sent out. That's what that looks like. And then they converge all onto this area called the cerebellar nuclei. And then from there, those neurons actually send another axon to the entire brain. So there's convergence from the Purkinje cells onto the cerebellar nuclei, and then there's divergence to the entire brain. And most of the regions in the brain are structured like that, between communities of cells that converge and then communities that diverge out. Okay. Next, I'd like to talk about a different brain region, and this is from the brainstem. It's called the LRN, or it stands for the lateral reticular nucleus, which sends a cell into the cerebellum like this, an axon, and it talks to this granule cell, which then talks to the Purkinje cell. So what does this system do? This system enables us, oh, okay, here we are. This is what they look like in the lateral reticular nucleus. These are the lateral reticular nuclear neurons, which I labeled in another transgenic mouse. But these neurons are important for reaching. Without these neurons, we would not be able to reach for a cup of coffee. They are about 8,000 of them, and they are in this brain region that looks like a shoe horse. Shoe horse. Yeah, I think that's what it's called. And this is what they look like. So they're blue here in the image, and their axon sends these highway projections, and then they branch out, casting this net into the cerebellum. So typically how it happens is you get an idea that you want to reach for your glass of water, and that sends a command to neurons in the spinal cord. And these neurons have a bifurcating axon. One axon is going to speak to the motor neuron, which is going to enable you to move your hand. And then the other axon actually goes up into this region, the LRN, lateral reticular nucleus, which are these blue neurons. And that speaks to the cerebellum. And what that does is it makes sure that you have a very precise ability to grab that cup of coffee. You typically don't miss when you grab your glass of water or your cup of coffee. You do it with perfection, and it's because of the system is there in place. Now, if you watch a child who's a couple of months old, and when you watch them reach, they miss the target object because these pathways are not laid down just yet. All right, so now I want to show you again this convergence concept that I was talking about. So Purkinje cells, I think in the human there are about 500,000, and again a lot fewer cerebellar nuclei, but you have a massive convergence of the Purkinje cell community onto the cerebellar nuclei, and then a massive divergence. And here's what the divergence looks like. And just to give you an idea of now we're in the millimeter range. Now we have to have different kinds of method to be able to start at one of the cerebellar nuclei and track one long axon to see the highway. Because if we understand what brain region is connected to what brain region, if we understand that the highway is there, then they're probably talking to each other in some way. So I make a little injection with this black dye, and then I make a very tiny injection. This is, I do it through neurosurgery, and you can't stain too many neurons because then you're not able to follow a single axon. Then I follow it in each slice. So from the beginning to the end, we actually have about 80 slices here, and we can see that the axon bifurcates into the superior colliculus, which is important for your vision, your eye movements, and then also into this area of the thalamus. So now we know these areas are talking to each other. And I want to show you that this is what I see in a typical slice, but I have anywhere from 80 to 100 of them. So sometimes I get to see a very big piece of axon like this, but other times it's just a little tiny piece that I see in that image. And I have to piece it together with the previous image and the next image. So it's all serial imaging reconstruction that we do across 100 images. And then I put this whole puzzle together to understand what brain region is talking to what brain region. And then we map it out, and now we understand the structure, and we begin to understand that the cell here, number 1, is talking to this area in the thalamus, and so on. You get the idea. We map it out, and this is how we begin to understand the function just from structural studies. Now I'm just going to show you some more beautiful pictures of the brain. These are a few other methods that I use. We're getting more and more sophisticated. We're using better viruses to deliver fluorescent proteins. In this case, this blue fluorescent protein. It's actually green, but I made it blue. Is delivered to one of these neurons. And we actually also have a marker. This red cell is a cell that was activated by an environmental stimulus. I actually gave a drug, a morphine, to this animal. So we can begin to see the patterns of cells that are activated. For example, here, you're looking at 2 cells that were both infected with a virus in a different brain region. So this tells me that these cells are connected to another brain region far away. And because they are yellow, the green marker is actually an environmental stimulus. It's a morphine injection that I gave. So now we have cells that are activated and connected to this other brain region. So this is how we're beginning to parcel out and bringing some information about activity of the neurons. And this is those pyramidal cells that Cajal was talking about. He called it the psychic cell. They are called pyramidal cells because they have a pyramid-like structure. And these are in the cortex being the region that is evolutionarily the latest. And also in humans, the cortex has greatly expanded compared to other animals. These are two cells. They're both infected with a virus. The yellow one is activated by an environmental stimuli. And this is a zoomed in photo. And when I say cells, these are actually neurons, to not confuse them with the glia. So this is a transgenic mouse that has red in a specific cell type. It's it's always red, but then you can inject the virus and you can begin to study some other neurons and also their projections here. These are the axons, which are coming out of the cerebellum. This is how they come out, and they project to the left. They go to the whole brain, and you can see how they are branching out. And here, these ones are going to the spine, to the brainstem and spine, probably brainstem, I'm sorry. So now I want to talk a little bit about how we believe the brain works now. Traditionally, it was thought that every brain region had a specific function, and that's still true. But there's another layer of complexity that was theorized in the 1950s, and now we are beginning to see it experimentally because we have the methodology. So the hippocampus is known as the GPS center. It's what helps us remember how to get home. The basolateral amygdala, or just the amygdala in general, is always known as the emotional center. It can process emotions such as fear. Then you have olfactory, is maybe another brain region that's important to detect this smell. Thalamus is it's a filtering system, and then the cortex is that part of the brain that it's your executive function, goal-directed decision-making brain area. Nucleus accumbens is a big one in my field of drug addiction. So here's how we believe it works. So when we are, and this is just to give you an idea of how even the neurons are so connected with the environment. There's an important interaction that I want to highlight here. When we smell a beautiful rose, there are a group of neurons, not all 100 billion neurons become activated, but a select group of neurons becomes activated to interpret that smell. And I'm sure in the olfactory system, which processes smell, are going to be neurons that become activated, but also in the hippocampus, because you have to have some information about, am I in the rose garden and which country am I in? So some kind of GPS information. And there might be some neurons that are elicited just from this rose in your emotional center. Maybe you feel very pleasant from smelling it. So there's a sparse code that elicits an activation of specific neurons in response to certain environmental stimuli. Maybe you meet someone you like, that elicits a different kind of pattern. If you inject heroin in the body, that will elicit, just the sight of the syringe will very likely activate a specific set of neurons. If you experience a traumatic experience, that will also elicit an activation of another set of neurons. And that's how we believe, and we're beginning to show this experimentally, that this is the case indeed. This is how we are able, through our sensory systems, to interpret the myriad of environmental stimuli around us. And of course, I'm going to mention something about the church now for the Orthodox friends. So the Orthodox liturgy is like a contact sport. Many describe it because it elicits basically every sense. So you have the Eucharist, which will probably activate certain neurons definitely in the brain area that processes taste. but also smell from the incense, or perhaps the chanting will also end up activating a different group of neurons. So you have this, very, how do you say, very full experience when you're activating all of your sensory systems. And here's how we can do that. So typically, this is just an image of the brain in the amygdala, that emotional center. And thanks to a marker, so if we present an environmental stimulus such as a rose, we now have a marker. It's called CFOS, and CFOS is a marker for cellular activity. It tells us, it elicits a gene expression and produces the protein, and it tells us this cell is now active. And that's been shown and validated electrophysiologically. So this is how we now combine not just the architecture of the brain, but also we begin to ask functional questions. And through this marker of cellular activity is how we can connect the behavioral response and environmental stimuli with what's going on in the brain. So the complexity of the brain does not only depend on its architecture and its structural elements, but rather on this immense capacity of interactions. This is the point that I wanted to make, between neuron to other neurons, neurons to glia. Neurons and glia actually talk with each other. Glia provides support for neurons. There's probably also glia-glia interactions. Glia bloodstream certainly interact with the vessels. There are neurochemicals which I didn't really speak about, such as you might have heard about dopamine, oxytocin, these ones. All of those are there as well. There are also electrical properties of neurons, and then you have, neurons that become activated by environmental stimuli through the sensory system. So there's a whole lot of interaction. I think that's really the most spectacular thing about the brain. So this interaction doesn't exist just in the brain. It actually exists in other parts of the universe, such as with the tree root system. There are some studies, and I think they were sensationalized a little bit, that trees talk to each other like human beings. But, without being, how do you say, without giving into these sensationalized stories, the truth is that there is some interaction between the tree roots underneath the ground. And then there's also all of these fungi that form networks underneath. So this is where other scientists as well are talking about this idea of why relational interactions and relationships are important, and they are key to existence, and they shape reality, whether we're talking about two subatomic particles or human to human interaction. And interactions are thought, what I believe as well, what gives rise to something, to the existence of something. So we're thinking now not so much about how things are or what they do, but really how they affect one another. And we're really thinking the reality in terms of relations and interactions instead of objects, entities, or substances, like the example in the brain that I said. That really the amazing thing about the brain is the capacity that it has for interaction. And then I'm switching now a little bit to this central theme of Trinitarian theology that I love Catherine Lacunia's discussions about it, in terms of God's relationship with us and our relationship with one another. And I will dive a little bit deeper into this in the next few slides. But I just want to make a very kind of, before I switch, just a little summary that all phenomena in the universe are interdependent in a complex manner. And without this interaction and interdependence, nothing would or could exist. And there are other people who have thought along this way. So this is from Plato, the sophist, and this is published in this article of Science. Anything which possesses any sort of power to affect another or to be affected by another if only for a single moment, however trifling the cause and however slight the effect, has real existence. And I hold that the definition of being is simply action. And then also in this idea of emptiness tells us that nothing has independent existence. Anything that exists thanks to a function of or according to the perspective of something else. All right, so now I will put my theologian hat on and present this icon, which is the hospitality of Abraham and Sarah, welcoming the three strangers in the desert in Palestine. There is communion here between the three persons of the Trinity. But what I like about it is that there's also communion with the human beings, with Abraham and Sarah. And in his chapter, A New Heaven and a New Earth, John Chrysaviges makes the point that actually there's interaction with the environment, because if we read carefully this story in Genesis chapter 18, it talks about an oak tree. And now we have trees that are, providing shade here. So there's a lot of interaction really here. Okay. And so there's a lot of talk from theologians about the relationship between Father, Son, and Holy Spirit. And I really like Catherine Lacuna because she points out that theologians became increasingly concerned with how the Father, Son, and Holy Spirit were related to each other, to the point that the Trinity model became a discussion of the heavenly committee, separated from the people. And then I really love Dumitru Staniloae. I read a lot of his work, and he does a beautiful job as well, speaking of the perfect love between Father, Son, and Holy Spirit. But then he goes one step further, so he's able to poke a little bit of holes into this separation. He breaks down this separation a little bit between the heavenly Trinity and the people by saying that through Christ, you have this relationship of perfect love that extends now to the people, actually he says to the church, and that means the community, and he says therefore the faithful. He emphasizes that in particular, because of the incarnation of Christ, this perfect love between Father, Son, and Holy Spirit is now extended through Christ to the people. So now human unity, although we share imperfect love, compared with the eminent Trinity, is intra-Trinitarian, is the word that he uses. And he presents this idea of unity in diversity among the people, just like unity among the three diverse persons of the Trinity. So like this, Staniloae affirms a better link between the Trinity and the Church, and thus the people of the body of Christ, representing this Trinitarian relationship of love, albeit not a perfect love. All right, 1 minute. Okay, so Lacuna actually pokes even larger gaps in this separation between the heavenly committee, interaction of the three persons of the Trinity and the people. She says that the encounter with God of Jesus Christ makes it possible for both our union with God and communion with each other. Christ penetrated humanity through the incarnation, and thus now our communion with God is enhanced, but also our communion with each other is enhanced. So she adds that part more clearly, that our communion with each other is enhanced. She more effectively eliminates this division, in my opinion, between the eminent Trinity, which is about God's inner life, the Father, Son, and Holy Spirit, and the people, by focusing not only on the relationship with God, but our relationship with each other, between each other. And I think that's so beautiful and brilliant of her. She goes one step further than Staniloae, in my opinion, to say that there is more than just unity and diversity, but an actual communion and relational interaction. Well, she highlights that relational interaction a little bit more for me in her work. Okay. She makes clear that the Trinity is a model for God, for who God is, a revelation of God in Christ. Because of this, God's life is shared with us. Living a Trinitarian faith means to live as Christ with total confidence in God as a healing presence. I'm just sharing with you some of the ideas from her work. So this way, the Trinity is not an abstract concept anymore about God's inner life in heaven, but helps us articulate how personal conversion is related to social transformation, including what constitutes the right relationship with the Christian community and society at large. It also helps us better understand the gospel's demands and how to best praise God. And she actually says that this is the liturgy shapes Trinitarian faith, especially in the sacraments of baptism and Eucharist, and recitation of the common creeds. She makes a beautiful point to understand that the relationship with God and with each other, since all human, that there's a relationship with God, but also with each other, since all human beings are made in God's image. All right, so, moment. Excuse me. Okay. At this point, I want to take one step further and advance my own thesis to say that the human-to-human interaction with each other can also shape Trinitarian faith, even outside of the traditional sacraments, which Lacuna says, they are the baptism. I think it can be outside as well. So human-to-human interaction gives rise to existence of a reality and provides an opportunity for divine revelation. In this way, the concept of the Trinity, whose goal is to try to understand who God is, flows out of the religious institution to all people. And if we focus on this concept of relational interaction and communion with each other and the environment, I think we can encompass more people. So what I try to do really here is push the doctrine of the Trinity one step further from Stanislaus' unity and diversity among human beings, among the faithful through Christ, and Catherine Lacuna's communion with God and each other, to say that it is the interaction with each other and the environment that opens our consciousness to the existence of divinity, of what is sacred and holy. I think that is the revelation. And it's because of this relational interaction that exists at every level in the universe is what gives rise to understanding the existence of something beyond and what most people call God. Okay. I will finish almost with, right before my conclusion, with this slide, and to say that God had, for lack of better word, and I'm sorry, I know I'm Orthodox and I'm supposed to think of God the Father always, but the more I study theology and the more I combine it with science, anyways, I will say God has always been, there's been holiness, sacredness, divinity, always irrevocably bound to people in our history, even our pre-human history. As you can see here, these are the last, 7 million years of pre-human and human evolution, and we are just here. But there's been a God irrevocably bound to people in the past and in the future, and there always will be. So maybe the new species, some people are saying, will be the Homo cyberneticus, which is this interaction, human AI interaction, to provide more augmented capacities for human beings. So with that, I will conclude. My conclusion is that the idea is that all phenomena in the universe are interdependent in a complex manner, and that without this interdependence, nothing would or could exist. And for me, I combine art, science, and theology to make visible what is previously unseen. So with the tools that I use that we have here in science, we can see in the brain, which is not available to the naked eye. We need the genetic tools. We need all the creativeness and equipment that we use. And then with art, is really good about highlighting the nuances, because in a lot of times the tissue that is stained, we have to interpret the salient features artistically. And then theology helps us go beyond when we have limits, certain limits in science as well. And with that, I'll end by saying that if we understand how things in the universe are interdependent, making these complex webs of interaction, then we can live more accordingly in respect to each other and the environment. So thank you for your attention. All right.
Thank you very much, Hermina. This was a tour de force, and you walked us through many places. We visited together with you all these places within us, outside us, in the heavens and below.
I like
This
that.
is yeah, amazing. Usually I say a few words before I give the floor to the respondent, but since I'm the respondent, I think I'll get right to it. I have a few slides to accompany my comments, but before I get to those, I learned something that I didn't know. Of course, it's not my field. That early 20th century discussion, controversy about the nature of neuronal networks. Are they continuous or discontinuous, with the two gentlemen, Golgi and Cajal? Yeah, this reminds me of another famous conversation of this kind that took place perhaps a couple of decades earlier, and was sort of sorted out in at the turn of the 20th century by Einstein with the nature of light. What is light? Is it a field? Is it a wave? Or is it an atom, or a body, a tiny body? And yeah, it's both. And I love this symmetry in neuroscience, as you emphasized it, that it's actually both. Yeah, the networks are continuous and discontinuous. Yeah, that's amazing. Plus, I thoroughly enjoyed that brief passage you presented from Cajal's Recuerdos de mi vida, which sounds like, a poetic depiction of the inner scapes, the mindscape. Yep, there you have it. Beautiful. And it reminds me of a similar passage in the beginning of a 6th century patristic work by John Moschus, The Spiritual Meadow, where Moschus describes the network of ascetics in Palestine, actually, as a beautiful rose garden. Yeah. So evocative, on many levels. Anyway, there's a lot of good stuff in what you said, and I thoroughly enjoyed this aspect of perceiving the Trinitarian nature of reality as the internal pattern of all creation, hence this kind of natural holiness, although that's not the phrase you used, that should invite us, encourage us to appreciate better all of the history of the universe, history of life on Earth, including the history of our own species. On this note, I'll turn to my own slides. yeah, you have beautifully unveiled for us the invisible side of reality as a way of perceiving reality as connectivity, or as you prefer, convergence. A reality organized around Trinitarian patterns of fellowship and cooperation. And I think you made a very important point, theologically speaking, because not many theologians go beyond this aspect of, okay, so what we learn from the Trinity, that we should cherish both the person or the persons individually and their relationship. It's more than that. It's the idea of this belonging together, this interaction that walks, if you like, across the spectrum of reality, uncreated and created. And a theologian, a Calvinist theologian that I thoroughly enjoy returning to from time to time, Jürgen Moltmann, says something along these lines, you know, for a Trinitarian perception of creation in his famous book, God in Creation. Every monad has many windows. He has a go here at Leibniz. Leibniz, you know, the modern philosopher who said, well, everything is a monad, so like an atomistic kind of model of reality. And he says, well, the monads have windows. Every monad has many windows. In actual fact, it consists only of windows. All living things, each in its own specific way, live in one another and with one another, from one another and for one another. It is this Trinitarian concept of life as interpenetration, or perichoresis, the Greek concept, which will therefore determine this ecological doctrine of creation. And this is a fundamental concept for his worldview, for Moltmann's worldview, which resonates very deeply with me and my perception of what a true Orthodox, Christian Orthodox view of reality might be, if the Orthodox will learn a bit more about their own tradition. Your paper is so significant in that it helps us understand how we can bridge distinct worlds, and the fact that we have to bridge these worlds, scientific research, theological insight, including artistic expression. And I was actually disappointed because you didn't mention the fact that some of the images that you presented were your own visualizations of what happens within the brain.
Yeah, they were all my own.
All of them. There you have
yeah,
it.
with the exception of Cajal's, the dark one, and then some of the 19th century ones, but all the rest are my own.
Wonderful. Thank you for clarifying it for us. And yeah, the fact that we need this kind of bridging the gaps between different aspects of our experience, fields of study, and so on and so forth, in order to establish a worldview that is holistic, comprehensive, and relevant existentially, culturally, and of course, socially. And it's important to point out the fact that this discourse of yours originates in your manifold training and formation as a scientist, a theologian, an artist. I'd like to point out the fact that this approach...
I'll just add one more thing. Can I just add one more thing?
Yeah, go on.
Actually, most scientists are artists. Most scientists are artists, because we have to create experiments. Very similar to how artists make a canvas. So there's a big creative factor that that scientists have to have. I just wanted to say that.
Yeah, well, and this shows how archaic and traditional scientists are, because this is what from the outset happened in the history of humanity, in the history of culture, where all those scribblings on rocks, caves, and all that were both art and science, and I'll get to it in a moment. So it shows that there's this perennial drive motivating our quest for knowledge and understanding that pushes us towards bridging the gap or the gaps between various aspects of our experience. And this is actually what Tom McLeish says in a recent article, 2021, The Rediscovery of Contemplation Through Science, where he says that the task of our time goes as follows. We have to work towards reorienting science from the idolization of pure objectivity to a duality with immersed, incarnate subjectivity, from a false notion of pure rationality to a recognition of the partnership reason has always enjoyed with imagination, hence art, yeah? From an exclusive expertise to a shared participation, contemplation, and joy, and from a convergent and prosaic narrative of the material world to an open, poetic, and infinitely beautiful one. And as a matter of fact, it's not only scientists who reach out artistically towards understanding and towards others, or their audiences, in order to communicate in a cogent, intelligible way. This is what theologians have done, and I have a few examples to present in the next few moments. Let's not forget the fact that many theologians of past ages were also great poets. Some of them were iconographers, and my examples are from this area of iconography, but many of them were poets. The best poetry can get, for instance, in the 4th century, first half of the 4th century, comes from the plume of Saint Ephrem the Syrian or Assyrian, who preferred to express himself rather poetically than prosaically, yeah, and so on and so forth. It's important from my viewpoint, and this is something that you highlighted time and again throughout your presentation, or towards, anyway, towards the end of it. In the words of Arthur Koestler, it's important to band together the mystic and the scientist to be able to look at reality from different viewpoints, the more rational, experimental, or experiential approach of the scientist, but also from the insightful views of the mystic. And in a way, this is what the traditional cultures of the past had always attempted to do. From the very first undertakings to enshrine their observations of natural phenomena into what, for want of a better word we call rock art and cave art, because there are doubts as to the aptness of this vocabulary. Is it art or not? And I am currently reading this book by Jean-Claude. What is Paleolithic art? And he says, well, we call it art, but is it art? Is it? Is this what they created? And actually, this goes well with my current project into zodiacal imagery. Yeah, well, have I told you I'm a perennial student. I'm doing a second PhD now in religious studies at the University of Sydney, and I grapple with archaeoastronomy leading up to the appropriation of the zodiac by Christians in the Middle Ages. Anyway, nice. This is just to give a few examples of how in prehistory, in past ages, people have always attempted to make sense of reality, including the starry sky, through, let's call it, artistic representation. You have all these cave paintings and scribblings and rock art and so on and so forth, which sort of come under this category of as in heaven, so on earth. I can't find better words, you know. And this is an undated depiction of the Pleiades, the seven sisters in South Australia, Walleninga. It's a beautiful depiction of the Pleiades and how significant they were for Australian Aboriginals. These are images that are fairly rigorously dated. Göbekli Tepe in Turkey, 11th century BC. We have a zodiac calendar. At least that's one interpretation of those pillars, that they aren't ornamental or placed there for some religious purposes. They represent a calendar. Of course, there's controversy around what this calendar is supposed to teach, but still, they were able to do that so many centuries ago. A more recent rock carving in Kaschena in Switzerland, 8th to 7th century BC, which indicate the pathway of the sun and stars. So these people were interested in the outer world. So what I'm trying to illustrate here is what you did from the viewpoint of your expertise as a neuroscientist. I try to illustrate by way of images of the macrocosmos, the universe.
Yeah.
And this is something, again, it's beautiful artwork with two ladies, one of them better dressed than the other, from the 16th century BC in Akrotiri, Santorini, where you can see stars. The universe is not deprived of the starry sky. And those stars are connected like Hermina's neurons. Yeah. So they had, we call them prehistoric people. Our predecessors had this idea of the connection of all things. Why those dots there? Do you tell me, what are those dots connecting the stars? Much beloved representation of animals at Lascaux. And according to the interpretation of, well, unfortunately, controversial author, not because of what she says doesn't make sense, but because of the rigidity of the established view, Chantal Volkiewicz. She wrote this book, which is a compelling analysis of Lascaux in terms of archaeoastronomy. And she says all those images of animals, especially when they cover the ceiling of the cave, are actually constellations. And she builds this compelling case for these animals as actually representing star maps, especially those dots. If you see those dots, yeah, they aren't just images or pictures of animals running. They actually again illustrate a keen observation of the rhythms of nature and how things unfold across the eons. Usually these cycles were either lunar or solar, but I don't want to get there. But what's important from my viewpoint, at least from the viewpoint of my current project, is that these animals, called by the Greeks zodia, gave us more recently in Babylon, the Hellenistic era, and we have here the famous Dendera zodiac of Cleopatra, who was an astronomer, a mathematician. She wasn't only the most beautiful woman of all times. She was actually a scientist, a philosopher herself, and those prehistoric zodia gave us this zodiac at almost the dawn of the Christian era, and upon this pattern, the Roman era came up with many more zodiacs, but I don't have time to get into that. I'll jump to Byzantine and post-Byzantine Christians who, surprisingly, against the anathemas of the early church centuries, reinterpreted the zodiac and appropriated it and made it part and parcel of their theological universe. And for them, all visible things conspire, cooperate towards revealing the invisible. Here's a passage from a 3rd century author, Origen, who in his commentary on the Song of Songs says something along these lines: from things visible, we can know the invisible things, from the corporeal, the incorporeal ones, and from the obvious, the hidden, specifically when we grasp the manner in which the divine wisdom established it. The creation of the world itself teaches us about the invisible things through visible things and images, transporting us from the earthly realms to the celestial ones. So the idea of Origen is that if we make sense of reality, the visible reality, and he didn't shy away from bringing in, let's say, the available sciences or the natural knowledge of the time, we do that in order to understand a reality which itself is, well, let's call it theophany, which invites us to contemplate the invisible. And unveiling the invisible was, for these early Christian and Byzantine Christians, a way of restoring the fullness of our experience. I have 2 more slides. This is a visualization of Psalms 148 to 150. It's post-Byzantine composition, 17th century, found in Svetitskovelik Cathedral in Georgia. Parts of it are lost, but you can see in that series of concentric circles, the outer circle is the zodiac, then angelic beings, and then verses around Christ's image, verses from the Psalms, and then the image of Christ in glory, who is the center of all reality. And all around this series of concentric circles, we have depictions of aspects of reality, cosmic reality, environmental reality, and so on and so forth, human activities, showing how everything for the Byzantine mind orbited around this image of Christ, or this revelation of Christ, which is facilitated by both visible and invisible realities. And I end with a Romanian depiction, an earlier depiction, 16th century, where the zodiac is part of the Last Judgment fresco at the monastery Voronezh. And you can see the zodiac, but not as a circle, just underneath the eave of the roof. In the center, again, Christ, but he's like popping up through a window with the 2, let's say, gates of the window, or whatever, coverings of the window being opened by angels. And on the 2 sides are 6 and 6 zodiacal signs, where, again, even at the end time, we have the presence of the cosmos, because this is the zodiac, but reorganized alongside this revelation of Christ, who is the center of all reality. And I believe that whether we are talking about the inner dimension of the neuronal networks or the macrocosm, it's legitimate for us Christians to incorporate that reality, that knowledge within our own worldview and communicate it artistically. You did so by way of your own artistry. In the past, we've seen people doing that in all sorts of visual forms, from rock art to church art, and so on and so forth. I spoke for too long. Yeah, I'm sorry for this. Let's take a short break, a 5 minutes break or so, and we resume with a Q&A after that.
Okay. See you soon.
We are back. It's good Hermina is with us, although it's so late in her part of the world. We are grateful for this. Okay, is there anything you'd like to add before we take some questions, comments from the audience?
I think we covered it all. I think this idea of focusing on interactions at all levels of the universe and how this reveals God is really the main idea.
Yeah, and well, you did so beautifully. You highlighted that aspect. I think I lost this topic a bit in my archaeoastronomical foray, but to an extent, I think I managed to highlight the importance of the topic, at least when I mentioned Moltmann's worldview, which is so inspiring still after so many decades. So that's a classical book, and I believe that
Yes.
that book is like an embodied Trinitarian theology that doesn't need to undertake Trinitarian theology in a technical sense, but it's all about the Trinity when it comes to talking about this world of convergence, connectivity, fellowship, interaction, cooperation, so on and so forth. And this is the revelation that the science, we expect science to bring to the world, because we still live in this paradigm, individualist, capitalist, and so on and so forth, where it's all about me, myself, and I. The idea of competitiveness and successfulness at the expense of our humanity. And as you said so aptly, our very nature, because as creatures, we are endowed with this internal, let's call it, pattern and program of divine fellowship imprinted everywhere by the Trinitarian God. Anyway, let's see if there are any questions from the audience or comments. There are some questions in the chat, but if anyone is up for Glenn O'Brien, Professor, please.
Thank you, Hermina, for a fascinating presentation, and just beautiful to look at those artistic depictions as well. I'm wondering if there's any, if your research connects in any way to certain pathologies, sociopathic behaviors, can these be understood as sort of there's something going wrong with the synapses within the brain? The neural networks somehow are failing or not firing, or think of a person who, a serial killer, for example, who doesn't seem to have empathy. There's a serious flaw in the connections between other human beings, and they act in ways that are very destructive and socially harmful. And we can think about that ethically and morally in terms of Christian, behavior and conduct and the doctrine of sin and all of that. Could there be, though, a physiological basis for these kinds of sociopathic behaviors? And is that connected in any way with your research?
Oh, what a big question. There are, you mentioned synapses. I'll give you an example of different disorders. For example, in certain mental retardations, the synapses are those protrusions on the trees that I try to point out. They're immature. They're like philopodia. So that's in the situation of a couple of intellectual disorders. Now, you mentioned about these like serial killers, or we would have to look at the brain. So the only way to answer your question, and the reason we know, such as in mental retardation, that those synapses are philopodia, is because someone actually looked in the human postmortem brain. So that's the only way that we can do it right now, at that level, at the neuron level. because MRI just, it gives us, maybe a bold signal where the blood goes to these brain regions, but it just doesn't give us enough resolution to answer your question. So those studies would have to be done in postmortem brain to see what dysregulation is there. But this dysregulation was not natural. I don't think, because the way it typically works is that the neurons and our connectivity in the brain is affected by the environment as well. Does that make sense? So because I want to be careful to not say, oh, my brain made me kill somebody. So that's, I hope I've provided, yeah, we really need to do more studies in post-mortem human. That's the only way to do it right now, because we simply don't have the equipment to look at that level in human. We're beginning to. Maybe you've heard, for example, that certain epilepsy patients have these electrodes deeply implanted in the brain. So now we're beginning to get activity. That's activity. It's not structural information at the neuron level, but it's activity, because neurons they fire action potential. So we can, neuroscientists can record activity from in these cases. I don't know if you guys know about this experiment. It was covered even in the New York Times. It was an experiment that was, it's not psychopathology, but epilepsy patients had deeply implanted electrodes in the brain to control their seizures. Okay. And then what they did, just to kind of tell you that we're getting there, we're going into that direction. But what the experimenters did is they played the Pink Floyd song to these patients who had the electrodes in the brain, and they were able to record the activity of that was being captured from the electrodes. And then using AI, they were able to reconstruct the song from the brain activity that these patients were listening to, this Pink Floyd. And that's publicly available in the New York Times. So we're beginning to be more invasive without harming the person. But to get to your questions right now, the only way to do it is in postmortem brain tissue. So if people with pathology are donors, then we can get those brains. In fact, I'm actually, I'm looking at the PTSD brain bank to get some brains and to study how stress induces PTSD. And then of those people who develop PTSD, they self-medicate. So we want to see what dysregulation happens at the neurobiological level. So we have access to these brain banks. I hope I have answered as much as I could to your question.
Yeah, no, that's great. Thank you very much. Thank you.
Yeah, it's a problem of we don't have the tools yet.
Okay, so because no one else raises the hand, let me read. Well, of course, you have some congratulations there. It's always good to see people appreciating the effort of our speakers. There's a question from a person named iPhone. I assume that's Nicola Hogarth Cregan from New Zealand, but maybe not. Anyway, so question: how does the structural knowledge of the microstructure of the nervous system up to gross anatomy provide insight into the functions and processes of cognition and emotion? What relation exists between cognition and emotions to the concept of the soul? Well, that's sort of outside the topic, but anyway, should this increase in precision about structure and process of mind and feeling change our comprehension of the soul, alter the definition of the soul with this new experimental evidence?
It's a lot of questions. So I know this is one of my friends. So, and he actually interviewed Tom McLeish.
McLeish, yeah.
Yeah, whom you mentioned. So there's an interview online about science and faith there. So I'll address the first question. How does the structural knowledge of the microstructure of the nervous system up to the gross anatomy provide insights into the right? So like in the 1500s, they were doing like Galileo Galilei when they, discovered the circulation of the blood, they were doing the big dissections. So that's gross anatomy. Provide insight into the functions and process of cognition and emotion. Yeah, okay, maybe I can give one example to that. Okay, studying the structure of the neural circuits is important. Let me give you an example. Oh, we've spoken about this together actually, Doru, and we can take the pain neural circuits. So before we were just evolutionarily, pain receptors existed before emotions. And so now that, we have more prefrontal cortex, those same neural circuits and a lot of how they are structured are also processing emotional pain. So it's the same neural circuits that once process like physical pain. So we know there's structure and that function, and then there's emotion that is now available, and that is also being processed by the same neural circuits. Maybe that's the best example. I can link the emotion to that. It's a hard question, this one. Okay. Yeah, the process of
I think
emotion.
it's really difficult to ask the question, about the interface of the brain and the soul and all that. I mean, that will lead you to a very speculative kind of area, and perhaps we don't want that. Any other questions or comments? Well, you have still time to make up your mind about asking Hermina your questions. Let me return to the topic of having science phrased out, spelled out as art. You mentioned the fact that many scientists are also artists. And yes, I have quoted McLeish saying something to that effect, that we have to bring together, to integrate all these aspects, facets of our human personality and experience in order to arrive at a comprehensive view of reality and a fuller experience of reality. I believe that it's quite interesting to get into the mind of scientists. I know how theologians work, theologians who become artists, but it's still enigmatic for me, to see how a scientist gets there. For instance, in your case, we're talking about your personal experience. How did it come to you? So this idea of visualizing artistically what you were observing under the microscope.
How did it come to me to okay, well, I mean, I just was captured by the beauty of it. And so I would go on the microscope, and then it would end up being 8 hours later, 4 a.m., and I'm still on the microscope, just looking around at all kinds of things. It's just so beautiful. It's such a huge universe that, I mean, I'm still discovering it. And
so this sense of awe, this sense of wonder at the beauty, the complexity of reality, this is what inspired you to become an artist.
Yes, because I thought I want to show this to other people as well, who might not have the tools, the equipment to be able to see this beauty. And even the pieces of art that I make, is just such a small window compared to the field of view that I have under the microscope, because what I can do is I can move around with the joystick all around the section of the brain. So I just, yeah, we're beginning to be able to, have ways to scan the whole brain. So we're getting there. We're getting there. But that's what it was. I thought it was so beautiful, and I wanted to share it with other people. So I thought to do it through art.
But is there a sense of, I don't know, some kind of mystical reverence, to paraphrase Einstein, at this beautiful complexity, this intricate and interconnected order of reality that sort of elicited this kind of response? Or is it just a matter of, well, it's aesthetically pleasing, and therefore I want to share this beauty with others? Is there
No,
something?
it's the first one. It's the first one because the structure has a specific function, which I was trying to get at. It's architecturally, everything in the brain is not random at all. There's a reason why the Purkinje cells are planar and flat. There's a reason for these convergences and divergence, how there are these situations in the brain. There's all a reason. And so then you connect it with, like I said, the cup of coffee. Now you know which neurons are important for that and how that works. And in fact, if you knock down those neurons, the organism, and I mean we do this in the animal, can no longer reach. You block that function at the behavioral level. I think, yeah, so looking at the structure and putting it together with the function is the moment of awe, if that makes sense.
But
And there's so much function that we don't know yet. So that's where Glenn was leading to, because a lot of, the movement stuff we know. For example, there have been so many experiments done. Gordon Holmes, I think, was one of them. He did experiments on veterans who were shell shocked, the back of the head, and they have motor movement. But now we're getting more complicated. Like in the work that I'm doing now, I'm speaking about emotions that drive people to take drugs. So it just gets a little bit more difficult. It's not like, let me knock this neuron out and now you cannot reach anymore. It just becomes more, I don't know how to say, the complexity is more, it keeps getting more complex.
Yeah. Got you. Thank you. It's always fascinating to see how, this deep dive in the mystery of reality, either via science or theology, mystical theology rather, elicits this kind of response, this sense of awe, this wonder at the complexity of reality that we mean to share with others, and you are doing it beautifully. There are a couple of questions.
Yeah. I want to add one more thing that as we were talking, even us as scientists, we actually, I don't know if this is the right terminology, but we make leap of faith. We don't, I mean, like in the 1950s, Donald Hebb theorized that the brain works with those, assembly of neurons that become activated by environmental stimuli. It's not until really recently that we are showing that that's true. He had no way of testing that then. So that was really ahead of his time. And Ramon Santiago y Cajal was also very much ahead of his time, probably about 100 years, because we are still answering some of his questions that he had from 125 years ago. So there's this leap of faith that we also make of how we think things work. And I think that's similar to people of faith as well, because there's this leap of faith that one makes. Scientists do it all the time.
Yeah, I think that you can call it insight or inspiration, and sometimes you don't know what to make of it.
Intuition, perhaps.
Yeah, perhaps. Yeah. Okay, so we have two questions that are quite related. One from my friend Martin in South Australia, and another one communicated by Brittany via Eastcast. Could Hermina extend her presentation into the integration of the mind and machines? And the other question, they are related. How might human and cybernetic interconnectedness give a greater capacity to comprehend nature as presented to us, whether in intuitions about the world, heightened sensory capacities, or improved general cognitive abilities? And before you turn to these two questions, I'd challenge Ted to step in if he wants to participate in this conversation. We had your maestro, Ted Peters, giving a talk last year, was it?
Well, thank you so much, Daru. Unfortunately, I tuned in late, so I don't have the facts to give an opinion.
But you have the questions there, and perhaps the two of you can have a go at these two questions that are related. So once again, the integration of mind and machine, if you can say a few...
Well, just let me mention one very striking thing, and then we'll turn it back over to Hermina. A couple of months ago, Hermina made a statement which has really stuck with me. She says she knows a lot empirically about how the brain functions, but there's a gap as you move from brain functioning to the mind. And what we all want to know about is the mind and the soul. And there's a gap there. And if there's a gap in science, how's the theologian going to interpret that? So hopefully you've already been discussing that, but if not, let's put that on the agenda. Thank you, Doru.
Almost, yeah.
Thank you, Ted.
Thank you.
And the other one...
Thank you, Ted. So nice to see you.
Yeah, so I was glad to see Ted coming to see his disciple performing.
Indeed. So what do we address first? Do we address the human AI?
Well, up to you. The floor is yours,
So
and we have a few minutes.
It's perfect timing because Ted is reviewing, or he's doing a second edition of his book. And so he invited me to write a chapter there where I address some of these questions. So could we go back to the actual question? I forgot what the person wanted to know. Oh, the interconnection, how the interaction between human and AI, and how does this increase interconnectedness? Wow. Okay, on so many levels that could be right.
Yeah. So there's on the one hand that there's the idea of connection mind machine or AI, whatever, and the other one is if this heightens our sensory capacities and therefore improve our, let's say, broader and more intense connection to the world, something like that.
Yeah, so that's something that I sort of demystify in the chapter a little bit, because we have these sensory systems, at least 5, okay, maybe 6 within in like other receptors on the organs that we don't really talk about, but we have sight, smell, taste, these ones feel. And if we are talking about AI or these machines enhancing our, I don't know how that would be able to enhance, because we still have our own human, sensory systems that we take in the information from the environment and it gets integrated and processed in the brain to make sense of it. But definitely there is the possibility to improve human life with certain tools that are being, AI tools or just technology in general. yeah, I just wanted to make that distinction. I don't know if it's going to enhance our maybe it does enhance our connectedness, being able to connect with each other like this through Zoom with this kind of technology. That's absolutely true. And I think in many cases, if you consider people who are suffering from debilitating diseases, you know, like with multiple sclerosis, very severe forms, when they can no longer speak, there are, you know, now devices that can measure the muscular movement, and people can do silent speech so that they don't put as much effort when they are, you know, in like a multiple sclerosis patient. May be able to communicate, you know, to project your voice takes a lot more effort than doing silent speech. So there's that kind of that can absolutely help and enhance somebody's life. That's one example.
Yeah. But you explained something before Ted joined us, something that I think is very important, at least for this discussion, this part of the discussion, the fact that the neurons somehow represent like an interface between, let's say, the inner self and the outside world, because you had those graphs where you showed how in certain circumstances, moments, situations, you know, either pleasure or pain or fear or whatever, anxiety, you know, various parts of the cerebellum, I think, are activated, the neurons in those parts. This shows that there's a mechanism put in place. It's natural. And the idea would be perhaps, could these AI enhancements boost our capability?
So that's something, again, that I addressed in this chapter. And it's so great that Ted had me write it, because what I didn't really go into at the beginning of the presentation is that neurons can fire an action potential, which means they can send a ghost signal to another neuron, or also fire an action potential and shut off another neuron. So they either excite or inhibit the partner that it's connected to. And then we have these global systems like dopamine system, where there's, or serotonergic system, where there is this net. You know, there'll be like dopamine system, you have neurons in the ventral tegmental in one particular brain region, and then it just sends these branches all over the brain like a net. And then from there, it pulsates neurotransmitters so that it's available to neurons. And those are called neurochemicals that modulate. But in terms of enhancing, now we have to think, how could that be possible if we put any kind of, a neuron can only take information of activity information in terms, like you can't, how do I say this? I think I explained it much better in the chapter, but a neuron takes in either excitatory or inhibitory information, and then it is modulated. And it's very limited. That's all it can do. So the idea of like, so you have these electrodes that could be potentially implanted into the brain, and that just either regulates the electrical activity of neurons. That's as much as it could do, and or modulate a little bit. But you can't like download a whole encyclopedia book through AI into the neurons. That's not possible. That's what I want to say. Or like download a new language. You know what I mean? Because there's a limit to the biological neuron. The biological cell has a limit to the way it can take in information.
So we stick as much as possible and keep maintain the system with which God/nature provided us.
Yes, there's still some limits.
Still, there's so much to know about it, to learn about it. Yeah. There's another question there, but please, Professor van der Hoek, John, can you please become visible out of your invisibility and ask your question?
Okay. I just ask you to comment. I just reveal the fact that I've been a cantor in the Orthodox Church for 40 years, and so the issue of training humans versus training AI is sort of an interesting concept, because the experience of being a chanter and chanting with that sort of experience, you experience psychological phenomena in the process of chanting, which it needs a long process of training to achieve. And I guess that it'd be interesting to know to what extent machine learning or AI can achieve similar things.
So while the question is, while you're chanting, there's a emotional involvement that takes place, maybe from
Yes,
the hymnography.
and you can't do it just by spending 2 or 3 years of training yourself in music. You can't do it.
Mm-hmm.
There's some interaction occurs between some very deep things in your psychology, whatever you want to put it, however you want to express it, and what you do. Okay.
Yeah, I think this is that gap that Ted so nicely talked about. We still have such a big gap between what we know the neurons are doing and then the behavior, these emotions that you're talking about. I don't have an explanation for how that works. Is what I'm trying to say.
By asking this question, I might just expose you to an area where it may give some insights.
Mm-hmm.
Thank you. I have a different kind of question. I'm very much interested in aspects of nature contemplation. And I wonder, Hermina, could you think of an example, kind of exercise that can help us look at the reality you are researching as a scientist, and build out of that ground a kind of contemplation of reality, of this internal reality leading up to the creator? Because this kind of exercise has been done many times over in the history of Christianity, based on the scriptures, based on the cosmos, and so on and so forth. But you have opened for us a window of possibility that I don't think it's explored.
So when you say this reality, can you on that a little bit?
The connections, the synapses between the neurons, all these aspect of, or landscape, if you like, of convergence and connectivity. Can it serve us, Christians, for example, to deepen our appreciation for God's creation and the creator?
Yeah, I think my observational experience as a neuroscientist definitely shapes my way of how I look at the universe, and why I keep saying that at every level of the universe there's an interaction that is critically important. Everything is, interdependent and codependent on each other, and it is this interaction that gives rise to something, to existence, and it shapes our reality. And that's all shaped because of my work under the microscope, and yeah, probably integrating with everything else I know in the environment.
Thank you very much. We are getting there. Glenn, another comment or question?
Yeah, I hope it's okay for me to have a second go. But just more an observation, but I'd very much welcome any comment that Hermina has. These interdisciplinary conversations are so important. One reason is Currently in the media space, there's a lot of talk about the rise of the robots and artificial intelligence taking over human beings and displacing them, and transhumanists talking about uploading people's brains to a computer by the year 2040, which is hopelessly optimistic. People kind of just take this in. But I find when I talk to people who work in artificial intelligence and in robotics, they're struggling to get a humanoid-like robot to pick a box up from one end of the room and take it to the other end of the room and place it on a table. Much less, like, that's not a robot that's going to overthrow the human race anytime soon, right? And then, of course, in your area of expertise, the idea of uploading a brain to a computer is clearly so far from the realm of possibility that it's ridiculous to talk about, other than for entertainment purposes. So I'm just highlighting the value of these interdisciplinary conversations for grounding things in reality by getting the perspectives of those who are actually the practitioners on the ground.
Yeah, thank you for that. I'm so glad that, Professor Ted Peters and Bob Russells and others have brought me to the CTNS in Berkeley and to communicate a little bit more like this.
Yeah, we have to wrap up. Thank you very much, Hermina. It has been a beautiful.
We didn't mention one last thing. Well, thank you guys. But I'm thinking I'm not that unique, because even Basil the Great, if we read his Hexameron, he made all of these observations in the natural world, and then he had reasons. Like, if you I read it a lot and I listen to it, and he said that the elephant, why are his tarsals fused? Well, he says it's because of to hold the big weight of the animal. It's just so great, because he observed and then he explained his observations. So I'm really just doing what other people from the 4th century were doing.
Yeah, and this is why I'm so interested in the topic of contemplation of nature, because it was done long ago. The history of Christianity is full of it, and I believe that with this new window that you have opened for us, we have more reasons to gaze into the mystery of created reality and learn to appreciate it, but also learn to appreciate the stamp of the creator into this complex reality. Now, before we wrap up, because we don't have time for more, unfortunately, I know that there's desire to have to further the conversation, but perhaps we can do that via email or something. I'd like to invite Professor Constantine Campbell, the research director of the Steering College of Divinity, to say a few words, because we are at the end of a 2-year cycle of seminars.
Thanks, Doru. I just want to add my thanks and congratulations. Hermina, thank you so much for your contribution today. I found it absolutely fascinating. And thanks, Doru, for moderating the session and for your response as well. My own research is in theology and also in the arts. And so the missing part for me is the scientific. And so to hear someone such as you, Hermina, bringing those 3 areas together in conversation was a true delight. And I think that in some ways actually is an expression of the very point that you're making, that everything is about our interconnectivity, and we're bringing together the sciences and theology and the arts in an interconnected way, and that's the way it should be. And so I think it's such an important conversation. And as this series comes to a close, just want to express my thanks and congratulations to everyone involved. So it's an important conversation that we must continue, and look forward to continuing in the future. Thanks, Doru.
Thank you, Con.
Thank you.
Yeah, our deepest gratitude goes towards Eastcast, our partner for the last 2 years. with these seminars. ISCAS has provided the Theology Research Network with logistics, with personal time, and I take this opportunity to thank Brittany Spencer, my ISCAS colleague, who has acted as a host for these last couple of seminars and prepared the ads, and will take care of the recording and edit and upload it in due course to the YouTube channel. It has been an interesting ride. I'm very grateful to Hermina and all the other speakers of this 2-year cycle, including Professor Neil Ormerod, who established the Theology Research Network and chaired it until a year and a half ago. And I'm very grateful to all of you for your patience, interest, attendance, month in and out. And on this note, let me close with my usual Digerigura Yana Janawi. In Darug, the Sydney language, means thank you for walking with me. And God willing, we will see each other in due course. All the best, everyone. Godspeed.